webinar. I have the pleasure of introducing our guest speaker for today, Dr. Shesh Rai. As the Wendell Cherry Endowed Chair in Clinical Trial Research, a professor of bioinformatics and biostatistics, the director of the Biostatistics Share Facility at the James Graham Brown Cancer Center, and the core director of the CIEHS Biostatistics and Informatics Facility Core, Dr. Rai provides support with design and analysis of preclinical and clinical studies. Dr. Rai has been recognized as a fellow of the American Statistical Association for his contribution to clinical trials methods. He completed his PhD in 1993 from the University of Waterloo in Canada and worked in the Canadian government before moving to St. Jude Children's Research Hospital in the year 2000. Dr. Rai is an expert with the safety issues in local and industry-sponsored clinical studies. He served on NIH review panels every year, and he serves as a statistical reviewer for many national and international scientific journals, including being an editorial board member for 5 journals. With his approximate 250 manuscripts and more than 35 NIH collaborative grants, he has advanced statistical science and collaborative research that deals with preclinical data, longitudinal data, clinical trials, clinical informatics, and retrospective studies and epidemiological studies. He directs multiple cores, including Biostatistics and Informatics Facility Core at CIEHS. So with all of his workload, he still enjoys teaching and has been nominated multiple times as a faculty favorite. His PhD and master students are doing quite well in pharmaceutical industry, government, as well as university settings. Currently, he's supervising multiple PhD students in biostatistics and bioinformatics. So Dr. Rai, thank you very much for taking the time to share your work with us today. And I'd like to go ahead and turn the floor over to you.
All right, that was a great introduction. Thank you so much. I so appreciate that one. Let me start, not sure. See if you all can see my slides.
Yes.
You can see my slides, right?
Yes.
Okay, good. So
actually, we just see you right now.
Oh, okay, sorry.
Slides disappeared.
Slides disappeared. Okay. Now you can see slides.
Yes.
Then if you click the full screen, then we'll be able to.
Okay. All right, right. Can you see the slides? This is.
No, down in the bottom, click the little camera icon.
This one?
There we go.
Okay. So thank you very much for the great introduction. I appreciate that very much. I am going to discuss the issues that I have faced in the analysis of microRNA data. So I'm not going to spend so much time on how the biological assays are generated, but just a brief introduction. I will talk about how do you define the threshold? How do you calculate the fold change? How do you find a housekeeping gene? And the sample size justification for the clinical studies. Then, if it's a longitudinal data, how do you do analysis? I also discuss about the normalization methods that plays a crucial role. How do you do the 3-group classification? 2 group is common, but what do you do with the 3 group? I talk about the prediction, and then briefly about the pathoanalysis, and then I also discuss about the issues in the preclinical studies that may be more relevant. So let's go ahead with as you all know much more than me, microRNA are non-coding RNA. And so increasing reports of they are generated from the multiple sources. The one issue is when the data is generated, there could be different sources of variability. So we are here focusing on what kind of source, what kind of variability affects on the defining the groups, finding whether this microRNA is significantly different than that one between the two groups. So how do you look into the batch effects, the group effect, and many other effects? We sometime back we published a manuscript, essay reproducibility in clinical studies involving plasma microRNAs. This was some time back. Still, this is very relevant. So I'll discuss some of the findings in this. So as we said, we are going to talk about the varying threshold, batch effect, operator effect, normalization methods, choice of housekeeping gene, methods of the fold change, and the group classification. So we are going to discuss in all of these things with examples. You have biological samples, the tissue and the plasma. Most of the time, in our experience, the tissue, you have a lot more expressions are there, the plasma less, more variability in the plasma, but this is how the samples are generated. You can generate the data in the different other assays as well. Let's move directly to the statistical analysis. As we said, the cycle threshold, statistical analysis, and reproducibility. These things plays a crucial role. Fixed threshold versus variable threshold, should you do pair t-test or should you do something else? Inter-operator comparison, how do you do intra and inter-operator variability? Now, going back to the threshold actually. Many times when you get the data from a lab, they give you optimal threshold value or corresponding CT value. What happens is, although the intensity, when you have the cycle here, and here is the log scale intensity on this axis. What you see there is, for a single microRNA, you may not get such a smooth, but hypothetically, you can get something like this. When the machine generates the optimal threshold, is picked somewhere around this, the red line actually. It picks you, gives you value. So that's corresponding threshold. Corresponding threshold is 0.8 here, something like that. And it gives you the corresponding CT value. Now, if you rely on the machine, it gives you always a varying threshold value for each microRNA. So that could become an issue. The one thing is in the linear phase actually. In the linear phase, you can pick any value, but if you fix them constantly the same value, you can compare from one group to another group, one microRNA to another microRNA. So we discuss about how do you choose what should be the optimal threshold actually. Should this be varying? Should it be constant? So your reliability is in the linear phase actually. You can have anywhere little less than the median value here or here. It's almost you get the same results. If it's less than this one is somewhere here, it's not very reliable. Now, I'm going to discuss a couple of data sets that we have used to demonstrate the lessons that we have learned. So first data set is CAA versus control. In this one, we demonstrate, and all this one is coming from the Dr. Susan Galandiak's lab, and she is expert in the colorectal cancer. So the data, the first one is we are comparing with CAA versus control. We show the effect of the threshold, variable threshold, batch effect, and operator effect. You may be thinking that there shouldn't be any batch effect and operator effect, but there are, and that plays a role. Then the second data set, we have a multiple groups: control, CRC plus CA, breast cancer, lung cancer, pancreatic cancer, and then we are going to discuss the different things in this data set. So first one is, if we were to compare between the two types of cancer, or between these three types of groups actually. Then you have a normalization. You could do quantile, you could do just delta, you could do the mean one actually. Another issue is selection of the housekeeping gene. How do you pick a good gene? For in this data set, we suggested these two. We'll show you how did we get these two. Then you have, based on this one, then next is the p-value, fold change, and there are different way of calculating the fold change, which is the most robust method of calculating the fold change. We will discuss in the thing actually. And then, as we said, we are going to talk about the 3-group comparison. So basically, quantile normalization, mean normalization, delta normalization, the delta method. These are 3 normalization procedures that plays a very crucial role, and housekeeping gene plays another crucial role in the data analysis. So what we say here is you could fix 0.01, 0.03, 0.05, or 0.5, something like that actually. But as we said, if it's in the linear phase, you pick it around that the middle or just above the little bit above the middle. But make sure that whatever you pick, you have a less missing actually. If so how do you choose this one? It should be in the linear phase and the missing should be minimum. This is how one should choose. Let's see. So then we have, and once you choose the threshold, you have a linear regression. So linear regression, you could do the ignore the threshold. You will have, you just fix a model like this. Delta CT is, this is a group effect. Whatever be the, whether the random or fixed threshold. If it's a fixed threshold, doesn't matter. If it's a variable threshold, it does matter in this model. But this is, so if it's a variable threshold, this becomes not reliable. Another option is you have a group effect and the threshold effect. Actually, you can have like this. This is very uncommon. And then what you could do here is delta CT. You use the threshold to use the normalization. So this is what you see there. You have delta CT. You have the CT of a particular microRNA, and then difference from the housekeeping gene. And then this factor is the varying threshold. So you could define something like this, and you can. So this is how you correct for the varying threshold. Okay, so now one thing is if you ignored it, here the microRNA that the focus were to study. Now all of a sudden you find a lot of them are significantly different. Is it the false discovery or true discovery? If you ignored it, you got quite a few of them. As a covariate, there's some changes, but still you get quite a few. If you use in the normalization procedure, now you see something is highly significant here. microRNA 142, 3P, now it's no more significantly different. The microRNA 27A, significantly different, all of a sudden it's not significantly different. The microRNA 21, not significantly different. And microRNA 218, not significantly different. And by the way, N1, because we are comparing the control and CAA, N1 and N2, we have a large sample size. It's not really small sample size. It's relatively good sample size actually. Now what we do is the same results if you want to see the group effect, and then we see if you have the fixed threshold versus variable threshold. Now, something is significant here, something is not significant there. Now, if I were to adjust for the FDR correction, this may not be significant and this may remain significant actually. So results are different. Which one should you choose? That becomes a challenging issue. Now we are more interested into this inter-operator variation data actually. But first we are looking for the group effects actually. Group effects, you have a fixed threshold 0.03, variable threshold. Just you use the variable threshold, we are comparing the two groups, the controls and CAA, two groups we are comparing. We see quite a few microRNA change the signs. Which one should we test? That becomes an issue actually. It could be because in this the group effect is there could be other variability in the data. That's why the results are not really consistent. So look at we have a threshold 0.03 and the variable threshold. Now here we have a group effect and operator effect. Now all of a sudden there is a operator for this microRNA, there's operator effect there. It's a significantly different. Here, variable threshold, if you use the model, there's no operator effect actually. If you use it for the microRNA 27A, its operator effect is not there, and then variable threshold, you have operator effect actually. So for this another one, 485, 3P, you have a very large operator effect, whereas you have a somewhat small size. For 21, operator effect is the variable threshold model, there is no operator effect, and then you have all of a sudden operator effect in this case. So that becomes kind of throws a doubt. We all want to see two groups are significantly different. For all these microRNAs are significantly different between two groups, but because of these operator things, it throws a doubt. Okay. Now then there's a batch effect. That means things are done in the different batches. So see what happens in that case. So we have a group effect, gender effect, and operator effect and batch effect. So basically now we are doing but this is for the fixed threshold. Fixed threshold, we have it there. And then what you will see there is the microRNA R21. The group effect is significantly different, but you have the batch effect also is significantly different. And on the side, the operator effect is significantly different. Now, number of missing within operator. So within operator, we have different. Here we are telling the 1 imputed, and here's 2 imputed actually. So that means how many out of the 16 and 16, how many we have to impute? For this microRNA 376C, we have quite a few of them are missing actually. And then, so the missing also with the different operator using it, we get even the threshold at the 3540, you do not get anything actually. So then we have to replace with the 40, so we have to impute. So that becomes the issue with the different operator as well. So basically we are interested in the group effect. The main focus is group effect, right? We use the fixed threshold. Fixed threshold, group effect, the p-values, so much change actually. P-values are changing so much because there is operator effect and then there's a batch effect. You put these things there, you have the p-value you are calculating. Without these two, now you have a p-value change so much for all these microRNAs. So that's about if you generated a data, that generated data has a batch effect also, and also has a group, the operator effect, then you should not ignore it. You should put that one in your ANOVA model or in your repeated measures analysis, and then you should claim that what are the what are the microRNA that's a significantly differing between the two groups. Now, there's so many ways people calculate the fold change. So this one is, the first one is, see, we take this, all the CT values, you sum it up, delta CT values, sum it up in one group, and then in the other group actually control. So this is the cases, this is the control, sum it up, do 2 to the power minus. So this is a one fold change. Now, when you, and then if need be, you can define the log actually, log of FC, so you can define the log FC. So you take the log of this quantity that gives you this. Another one is, you could have this one, the 2 to the power already done here, and then you sum it up for this. So this is more like a expression data actually. Expression in one group, mean expression is one group, and mean expression is another group. So that's another form that you can use. And then another case is, this is expression, but 1 over expression in one case, 1 over expression in another case. So this is kind of a, this is the expression based on the mean expression. This becomes more like a, this is a harmonic mean kind of things that you calculate actually. So again, this is another way people calculate. Now our focus is basically, the first one is most reliable. I mean, most reliable means they give you different values, but we suggest using the first one. Let's see how it affects actually. So the first one, for the microRNA, this is highly significant. One case is 1165, one case is 2000, one case 750. Now here is another one. If you're just, the first one is this one is upregulated, this one is downregulated. You see these values, although it's not significantly different. And here in this case is 0 and 0. So it becomes a totally different, the direction change actually from this one to this one, as well actually. So our focus based on our experience has been, go with this flow. This one is better choice. Now, the next thing is, how do we pick up the housekeeping gene? For the plasma housekeeping gene, first thing, it should be expressed in all samples, have a medium to high level of expression, have consistently measured levels of expression. So here we are suggesting these two, but I'll give you some examples why we are suggesting these two. So now you have I didn't discuss about the normalization method that I will discuss a little later. We have these, we are comparing CRC plus CAA versus BC, other type of, so colorectal type of cancer, and then all other type of cancers we are comparing here. Now, if you have one housekeeping gene versus another housekeeping gene, you have the U6 and you have a 525P. Now what you see there is, as we said, we'll focus on the FC1, so that don't worry on that one. We are more focused on the P values. So for 29C, it's a significantly different, and all of a sudden if you use the U6, it's become very highly significant. Then you have the R21. If you use the U6, it's highly significant, whereas if you use the 520P, it's a significant still. It's still for 19A, it's significant, but if you use the U6, it's very highly significant. And then you have the 372. It's a significant with this housekeeping gene, but it's not significant with the U6. And here the other change the story actually. So should you choose this? Should you choose this? That becomes issue. Once we use the quantile normalization, rather the delta normalization, results are similar, all over similar except for 372. Still the 372, it's a flipped. It's not significant here and becomes all of a sudden significantly different. So it becomes a issue, what should be your housekeeping gene? And I have suggested some ways to find a housekeeping gene. This is another paper that we published. So to find out the housekeeping gene, you find these are the potential candidates. So we have 6 potential candidates. Now in this one, all of them should be expressed. We said for these are the type of samples we have, all of them should be expressed. It's good to have if the threshold value, CT value is the lowest. But the problem is for the 484, it's the lowest in this case, but they have reasonably high standard deviation. So you do not want your housekeeping gene is kind of a variability is a constant across all these samples. It should be all these samples, variability should be kind of a constant. Now, once we look at the variability, the 520D, 5P, you see the variability is minimum actually among all these type of things actually. And then the next comes this U6. One could also choose, based on this variability, you could choose the housekeeping gene as the sum of these two, but in a ratio of the, what should be the weight based on the amount of the variability. Amount of the variability, you could have a different weight. For in this case, for the colorectal cancer, this is 3.8, this is 0.8. So I could come up with a weight to choose a weighted microRNAs to consider as a housekeeping gene, and weights can be not in terms of these the weight should be in terms of the standard deviations. So this is something very important when you are doing your study. You start with one housekeeping gene, all of a sudden you find that that is a lot more varying from your cases and controls. Then better you should look for other genes that's not varying so much, standard deviation is very small. Choose that one or a combination of a few to define your housekeeping gene. Now we change the gears because now our focus is statistical analysis. I will come back to the normalization methods, but now my focus is statistical analysis. If you do not do proper statistical analysis, you get the results that you really don't like. So this was some time back. We worked with Sumant Prabhu, and they had some idea that the microRNA in their repeat samples, they should be predictive of how the patient is doing. And they did themselves first page analysis. They did something, they didn't find it. Most of the time, the clinician, everybody try to use Prism, and the Prism, if they find something, they're very happy. They don't come to us. If they don't find it, they come to us, which is fine. So this is more challenging because this is a repeat measures actually. Samples are on the repeated times on the same subjects. So we need to data quality, we need to check the normalization, we need to use the proper method for analysis. So these 3 things are very, very important. This is what we talk about, some normalization here. So in that paper that I have cited, we have the detailed description. I'm going to discuss some of things here. So it's a delta method. Delta actually just take the difference, as simple as that. Just take a delta. So that means from the housekeeping gene, you have to take the difference. You are fine with that actually. Then there's a mean normalization. You have the CT value here. You take the mean over all the genes actually. You take a mean of it, take a difference. That's another way you could do it. Then this is a quantile normalization. Then there's a rank norm. I will explain you why quantile normalization is very important through example. Simplest example possible. I'll show you that. Then there's a rank based. Here's the artificial data. You have a 5 genes, 1, 2, 3, 4, 5 genes. You have the 4 replicates of the controls and 4 replicates in the treatment group. So you have these things are there. Now suppose these are the CT value. Suppose these are some expression value, CT value, whatever be there. I really don't care. Don't pay attention too much that these values are too small. These values, what's the range of these values are there. Don't pay attention on that one. This is just to demonstrate what the quantile normalization, if it's not done properly, makes a difference. Suppose the rep1 and rep2 are your exposed and non-exposed, are the medium exposure versus high exposure, or rep1 and rep2 within control, you may have males and females actually. So you have males and female, and the same thing here, males and females, and you see there is just some pattern here actually. First of all, in the controls, you see the values are lower, and in the treatment group, they are higher actually. Other thing you see, rep1 and rep2, this is slightly lower and this is slightly higher actually. Now the quantile normalization, many times what the software you use, they do quantile normalization is all over this one. They take one data set because you have this is one set of one replicate is there for all possible genes you are trying to do. Either you do scaling first, and based on the scaling, then you normalize for everything together. If you do normalize everything together, things becomes cumbersome. What happens is, if I did all normalization together, then you see there the clear difference that you have seen it before here, that difference disappears if I do everything together. So this becomes a problematic actually. If I knew, my focus is to find out if there's a treatment effect, then there's a treatment and control effect, and then also I know there's a gender effect also there. So then the normalization should be done differently. How should it be done? First of all, suppose I have only treatment effect actually. So that means these are the 4 replicates. This is controls, these are treatments. So you do normalization in this group by itself. Then you do normalization in this group by itself. Now you see each gene will be still significantly different. In other case, if you ignore that information, they will not be significantly different anymore. Now, as I said, same thing. If your focus is like not only one factor ANOVA, 2 factor ANOVA actually, and if you have enough replicates, then you do at the gender level also. So you do normalization, consider only rep1 and rep2 together, do normalization on this together. Rep3 and rep4 do another normalization there, and same pattern you follow in the treatment group. So this way you keep the same kind of the normalization. What it does is borrows strength from all the genes and the proper replicates. It doesn't borrow, it shouldn't borrow the strength from other replicate that you think that could be a factor for determining if which gene is significantly different and which gene is not significantly different. All right, so that's about the normalization. We have developed some tools in collaboration with Mike Merchant and others, and my students Srivastava developed for proteomics. We have developed a very nice tool that can do these normalization that we are talking about, gives a good result actually. Now going back to that, the heart failure data. So what happens if you do one-way analysis versus other-way analysis? There are some of the test procedures that I'm not going to talk about in details. Now, so once we did this filtering, actually, you have to do some data is not reliable. You have to do some filtering actually. You have data you have before filtering less than 35, CT value less than 35. You have quite a few of them actually, just only this percentage. After filtering in your data, you have a lot more data set is reliable here. And then greater than 35, CT greater than 35 is considered not very reliable. So you have only few of them here actually. And then same thing, if you have 3 group comparison here, you increase from 70% to 90%. So now it's good to do this one before you do your analysis. And then we did the quantile normalization as well in this. What we see there is, if you just did, see, you plot your all the p-values. And then what you see there is, if you just do the t-test, just do the t-test you're doing here, see, you have not too many things are significantly different here. Quite a few of them are here. So you're telling that they are not significantly different. We did the quantile normalization. We did this, the GE approach actually, which takes into account the same subject is their own control. So we have 2 groups, but the same subject is their own control. Do that one. All of a sudden, you have quite a few of them are significantly different. We are not going to talk about much more of analysis than this. The Sumant and we have a publication there, but this is just an issue. If you do not use the proper analysis, if you just do a 2 group comparison, you are going to miss quite a few actually. You did the proper, you use a proper method, you get quite a few of them. That's highly significantly different. So that's that. Now I'm going to talk about the sample size calculation. So that's if anybody has any question on this, the heart failure data, I can stop, take a pause and answer. Otherwise, I keep on going and then you can ask me questions later. So when you do the sample size, and I will come back, discuss something more on in the preclinical trials. Preclinical trials, what are the issues for calculating designing the studies? Very briefly, because that is itself a one big topic actually. So you want to have the 90% power, 90% you need it. You are doing the one sample size versus one-sided versus two-sided. FDR is 1%, 5%, and 10%. So these are the adjusted p-values. How do we get the adjusted p-values? Suppose you have a 400 microRNA that one could screen. Okay. Now out of these 400, you are interested only. You looked at the pilot data. You looked at the published report, and you found out of the 400, 40 of them are candidates actually. 40 of them candidates. Somebody has said that this one is expressed here and there. Now you have a 2 group interested in some kind of exposure. Now out of 40, you may be interested only 5 or 6. You need to define, you have to have a pre-hypothesis. And all 40 are not going to be significantly different actually. So you need to come up with the idea. Out of 40, if I find 4, I'm very happy about it. So you have a 1, 400 to 40 to 4. So that's a sequence is very important for you to have idea actually. And then you define a FDR. So based on this FDR, you use these adjusted alpha levels. This is not like the Bonferroni adjustment where you have 4 things you are comparing. So you divide alpha by 4 and then you do it. It's a different, actually, it's a totally different procedure. Now remember, if it's a one-sided and two-sided, the sample size is slightly less in one-sided. It's not whole lot. It's not whole lot because we do the derivations in my one of the class actually. Why one-sided versus two-sided? The sample size is not whole lot difference, but you need 18 and 20 actually. So this is the sample size if your FDR is 5%. If it's more like the clinical study. So if I have to choose the scenario I said, if I use the adjusted alpha this, I need 18 in one group and 18 in other group. Then what you need to do here is, suppose you found your microRNA to be significantly different. This is the this is what we do here is you need to take this the next step. What's the next step is you found is a 5 microRNA that's highly significantly different. Can this 5 microRNA predict which patient is going to fail and which patient is not going to fail actually? This kind of applications we have done with the Dr. Kelly McMaster and how. So in their studies, we have this one very often. Find out a set of microRNA that can predict. For that one, you need to have the power for sensitivity and specificity. So you use 10 microRNA. You need a 90% power. You need this many subjects actually. Here is a fewer microRNA. You need a less power. You need a less sample size. So this is how you do for the classification. This is for the finding the microRNA. And next step is if you find the microRNA, build a predictive model, either supervised or unsupervised the approach actually, and then how many samples do you need? Based on that one, you do discuss this one. So this is another piece in the clinical studies. Now for the predictions, we have multiple manuscripts and there are multiple references out there. Because if we have the simplest thing is the logistic model actually. You have a logistic model, you have multiple microRNAs are there, you have a delta, the things are there you are going to predict. So you can do this one actually, you can predict based on this one. You could also throw in the other covariates here too. So then you have based on just a logistic multivariable logistic model you can predict. Now the one thing is either use the logistic model or the probit model or some other models to predict. That's one approach. Other one is non-parametric model. You can use this. There are the different support vector machine and so many other procedures are there that you can use to predict. The one option is how do you say that I suggested that there are multiple microRNAs are there. How do I say that this one should be 2-fold, this one should be 1-fold, this one should be down-regulated, this one should be up-regulated. So that cutoff to come up with that, we need to find out what should be the cutoff probability. So based on all these ones, you come up with a cutoff probability to say that this is how it should work. The cutoff probability comes from the sensitivity and specificity score together. And most of the time you do sensitivity minus 1 minus specificity. That's called the J statistics. Then you define these two quantities. This is for the basic things that I'm not sure whether you really want to know. So this is how do we define sensitivity. This is how do we define a specificity. This is the positive predictive value, negative predictive value. Because still we are with that the 3-group comparison, so we are leading to that data set. We were able to predict with AUC, area under the curve, 0.97. That was very nice. Sample size was large enough. And the cutoff was 0.547. So suppose you got this, this is the ROC based on 1, 2, 3, 4, and 5. So there are 5 microRNA you use. You got the AUC is the like this actually. So now what you need to do here is find out one of these points that the J statistics is maximized. This one gives you the J statistics, that's the maximum. How do I get that one? For that one, you need to read this paper actually. And this paper is a very basic actually that talks about how do you get the J statistics. This has a good examples on how do you estimate the sensitivity, specificity, J statistics, all these things are there. Now we are talking about the 3-group comparison controls. These 3 groups, we are going to come up with that. So now in this one, you do multivariable, multinomial logistic regression to do it. Or you do the random forest. Unsupervised, non-parametric method, you could do this. What happens is, there's a, if you use a random forest, accuracy reached 100%. And the reason it reaches 100% because you use all microRNAs to predict. Now you can say, yeah, I'm very happy about it. Okay, but this is called overfitting. This is called overfitting because you use quite a few to predict something actually. If you, but I may want to know if I'm the patient, I want to know whether I'm one group versus other group, I'm control, are not in the control group actually. I want to see which microRNA meets the threshold. What's the threshold means? The threshold is, suppose I said it's a 2-fold upregulated or it's a downregulated. So and which microRNA? So it suggested the 5 microRNA and you have a clear pattern in those. Based on that, I want to do it clear cut. In that case, we have only 78% accuracy rate actually. In the logistic model, it's overfitting. You use all of it. This is the price we pay in the multinomial logistic regression. The accuracy rate is only less than 50% because it doesn't help much actually. It hurts actually if you choose. It's just like a regression analysis. You throw in quite a few variables, and that's not significantly different. It's going to hurt somewhere. That's why accuracy rate is this. But if you use most of the time AI techniques, the focus is increase the predictive rate. Okay, we are fine with that actually. But how, if I were to predict for a future patients, if it's the past data, I can have a very good predictive rate. But for the future patients, I need a specific thing. For that, multinomial logistic regression is the better approach. Pathway analysis, in our lab, we have a way to do pathway analysis also. We could do it. What we need to do here is we generate we do analysis, we take the fold change, we take the adjusted p-value, or if we have a raw p-value, we can calculate the adjusted p-value ourselves. We can calculate the q-value ourselves. We have the GNMIN actually. GNMIN is so good in this creating the small subroutines that can do the jobs very fast, very, very fast. So we can convert all these things and then we can put it in the IPA that we have a license. We can do it. We have done on the few analysis. So this is another thing that we can take the next step. I had one more slide. looks like I had one more slide that probably is, I was creating the last May. It's not here. Let me see where I can find it.
I think we might need to wrap it up.
Okay, just I just wanted to discuss a few minor things there. Just okay. So, can you all see this slide by any chance?
It's very small. Put it in show mode.
Okay. Can you all see this?
That's better.
Okay, good. So in the preclinical studies down the line, I will make another presentation sometimes. So now you have the outcome variable you need to decide. The continuous proportion and survival time. Suppose you are interested to the tumor size, shrinking the tumor size or some other the marker you have there. It requires a small sample size. You could get away with the 7, 8, 9, whatever the number you have the based on the preliminary data, we can calculate. But if you are interested into the proportion, actually, you are interested into the proportion that how many animals have improved and how many animal has not improved or something like that. If you have proportion in animals, then it requires a bit medium sample size. Now, if you have the survival time, that requires a large sample size. So most of the time people have this continuous in mind and they think that the 10 animals will do all the trick. No, it's just like you have to think of it. Is it the continuous outcome is primary and the proportional survival are secondary outcomes? Then you plan that way. But if the survival outcome is the primary, then you calculate the sample size based on that outcome actually. Adjusting the alpha. If it's a high throughput study, you are interested in multiple proteomics, protein data actually, use FDR adjusted alpha. If it's a selected, like some cytokines you are interested to study, then you could do the Bonferroni adjusted alpha you could do for the primary outcome variables. Also, when you design it, use a balanced ANOVA. If it's a 2-factor, make sure you have a balanced ANOVA. You may not want to do a mark and another control. You do not want to do the 2 controls actually. If there's a way, you just use only 1 control, balance ANOVA, but increase the sample size. I have seen the experiments people plan. They will have the 1 control, SAM, and then something else. So 2 control, why do I need it? Oh, just I need it because it's like I want to see something else. But there's no hypothesis for that. So it's better to have a balanced ANOVA approach and think of these things before you do it. Make sure when whatever you plan, always you have some animal will go wrong for one way or other. Inflate your sample size. Okay. And when it comes to analysis, use ANOVA and GLM model. Don't use a simple two sample test actually. Get the results based on ANOVA model. Use contrast to do things like that actually. So before I go, I just want to go back to tell my team. Here are some references. We just for the recently we published one of the paper, evolutionary bioinformatics about the statistical issues. I want to thank my team. They're in the different colors. So first one is the 3 in the red. My backbone, they are my staff. Shilpa has joined recently. So, and then here are my students actually. They are still very active, supporting and helping us writing these manuscripts, developing new methods. And then I have Dr. Park and Dr. Pal. Dr. Pal is his expertise is high throughput data and Bayesian, and Dr. Park is another expert in the bioinformatics tools. So we take help from these two folks as well. So that's my team. I would like to acknowledge all the center grants that they provide lots of support to us. I stop there.
All right, thank you, Dr. Rai, for that presentation. Now I'd like to invite the audience for a couple of questions. You can either raise your hand or send your question in the chat box.
I'll jump in. I was most interested in the analysis of operator variability. So we encounter this problem not just in microRNA analyses and RT-PCRs, but in lots of things. So how do we determine whether or not one or the other might be correct and not just rely on the things that are common?
That's a very good question, actually. If you think we need to check in the full ANOVA model, actually. If you think that the operator effect is significantly different, don't ignore it. Keep that one as a factor and use that one in the model. And then if the operator effect is significantly different, then that has to be mentioned when you publish your result. That this microRNA R21 we found to be significantly different. However, the operator effect was also significantly different.
Okay, thank you.
Do we have any more questions?
I forgot to add one more person, Christy Pinkston. She's another my PhD student working on, and she is developing quantile normalization and other normalization procedures for a different type of microRNA studies. And she is helping Matt Cave on his multiple studies.
Okay, so if there's no more questions or comments, we have a Cancer Rig meeting following this on another link. So the Cancer Rig members, please join us there. Thank you again, Shesh, for your seminar.
Thank you very much, Dr. Rai.
Thank you so much, everybody.